Documents in archival and modern shells from the Illinois River system. Dr. Aaron Wilson is an associate professor of environmental studies at Illinois Wesleyan University in Bloomington. His research has focused primarily on toxic trace metals in invertebrates from both aquatic and terrestrial systems. Pursuing his research objectives has taken him from the blackwater swamps of southern Georgia to shoreline of Lake Michigan to archives of Illinois Natural History Survey. Recently, he has also published on toxic lead concentration in ground venison from central Illinois. Let's welcome Dr. Aaron Wilson for the talk.
Awesome. Thank you very much, Tarun. Sorry if I'm messing that up. Thank you very much for that wonderful introduction. I'm going to go ahead and share my screen with everyone.
Yeah.
And I'd like to start off by recognizing my collaborators. So Andrea and Mark Fritz and Andy Casper were all at the Illinois Natural History Survey when this the bulk of these studies were completed. And then Jason Unrein at the University of Kentucky is the one responsible for most of the elemental analyses that I'll be talking about in this presentation. If anyone wants to ask a question, feel free to stop me. I just kind of tend to go on. But I was telling Torun that I'm going to be working on getting done in 30 minutes. So that's my goal. We'll see. So I want to talk about first where we're getting. So I'm trying to get data about trace elements from shells. So where are our shells coming from? So our shells are coming from freshwater mussels. So to give you a little bit of the biological background, if it's been a while since Gen Bio, then our mussels are mollusks, they're bivalves, and they're related to other types of bivalves that you may be more aware of, such as marine mussels, clams, scallops, etc. They've got a fairly simple physiology on the inside, but one of the things that makes them very interesting, besides their shell growth, right, is that their ecological niches in Illinois rivers is that they are benthic, they live on the bottom, and they're filter feeders. So they bring in water, filter out the particles that they can use for food, and then they expel the water. And so they are actually going to be exposed to anything going on in the water column and anything going around in the sediment, because they can also stick out their foot and sweep in some food that way. It's called pedal feeding. So they're going to be exposed to all of that. And this becomes interesting if you're a toxicologist like me and you're interested in the way that toxicants move around in the environment. And we'll talk about that in a couple slides. I also want to define trace elements. And what I mean when I say trace elements. Obviously, trace elements are defined by their concentration in the Earth's crust, but I'm mostly going to be talking about trace elements of toxicological and environmental significance. Once upon a time, these are called heavy metals, and then people pointed out, hey, there's not necessarily anything heavy about vanadium. Also, arsenic and selenium, those aren't technically metals. So now I just call them trace elements as a kind of catch-all term. Some of my colleagues like potentially toxic trace elements. I just think that's too much to say. So I've outlined the ones that are some of the elements that I typically study. And then I've highlighted in red 4 that I'm going to focus on for this talk, just because it's going to be too much to talk about all of them. So if you read my work, you'll see that I get into the nitty-gritty detail on most of the, several other elements, but I'm going to focus on manganese, copper, cadmium, and lead in this talk. Cadmium and lead are what we call non-essential elements. As far as we know, that there is no bottom to their toxicity. They are inessential to life as far as we can tell, and any amount of exposure to them is bad. Whereas manganese and copper are both essential for life, but if you get too much of them, copper more so than manganese, then you're going to have toxic effects. So the theory that I've studied and that I've been working with, it goes a little bit like this. All right, as these mussels grow, they have to grow their shell in order to accommodate their growing body. They do this in a seasonal manner because it gets cold in the winters here. It gets cold and dark, and there's not a lot of food. And so they kind of shut down for the winter. And so they tend to grow their shells in these discrete rings called annuli, very analogous to the rings of a tree. While they're growing these shells, they're grabbing things from their food to grow their shells, which are calcium carbonate based. Well, as they're grabbing elements, as they're grabbing calcium to make their shells, they accidentally incorporate other elements into the shells. If you look in the literature, especially elements that can get into the +2 ionic configuration. This makes, ideally, the theory here is that this would make the shell a form of what is called a biomonitor, a biological organism that is essentially testing or monitoring the water for these potentially toxic trace elements, for pollution. And this has been successfully applied, especially in marine systems. There's some really great work on intra-annual analysis, so analyzing those shells, those shell rings in marine systems. But in freshwater systems, it has just not been explored as much. We'll talk about that on the next slide. But the really cool thing is if we could get this going, if we could figure out how this works, we could use past shells that are dug up archaeologically, could tell us something about what was going on in the environment in the past. There's been some work with fossilized shells, or also shells that were collected in the past, that they've been some part of collection. We know when they were pulled out, we know where, and that could tell us also something about the recent past as well. However, a lot of questions remain. I mentioned freshwater systems. We are still kind of figuring out how this whole thing could work. Are the concentrations high enough? Are we going to have issues with detection? Ideally, a shell should be kind of a long-term pool of elements, right? So it's going to maybe vary less temporally than tissues. So we know we use mussel and clam tissues all the time to monitor elements. In fact, the US EPA has a long-running program called Mussel Watch. They sample mussels from the Great Lakes and along the coastlines, and they've been doing this since the 1970s on an annual basis so that they can measure what's going on with toxic trace elements and other toxicants in those systems. So we know that tissues work, but we also know that tissues vary very quickly depending on what's going on around them, and also reproductive status. So when you reproduce as a mussel, you lose a lot of mass because you're using a lot of energy to reproduce. And that when you lose mass, but the amount of trace elements stays the same, that means concentration is going up in those tissues. So we know that tissues tend to vary widely across in a seasonal way. But what about shells? Shells should kind of be storing this up. So are they a more long-term store? Are they less variable? Would they be better in that respect? But also, what other variables could influence those concentrations? One thing that I've done that I don't even have time to get into in this talk is I've looked at shell allometry, or the size and shape of the shells. Does that correlate? Does the growth of the individual correlate with concentrations? And I find out that yes, sometimes it does. But really, for our question about whether these shells are going to really tell us about pollution, really tell us about toxic elements in our environment, is the do they reflect what is going on in the water chemistry, what is going on in the sediment? Can they show us human effects, either indirect or direct from pollution or contamination? Are they then useful in that way? And can we use them as an archive for past conditions? So I'm going to go through a few slides, talk about how I've approached answering some of these questions. So my first part of my approach is a meta-analysis of literature that I conducted. Again, mostly from marine systems at this point. Freshwater systems have a lot less, just have been studied to a lot less smaller degree. And then what I'm going to mostly focus on in this talk is analysis of shells from the Illinois River and its tributaries. This was a variety of shells. So my colleagues at the Illinois Natural History Survey, they collected modern shells directly. So they collected living individuals and then sampled their shells. Then they also had historic shells with date and location recorded that they got from their archives stored on the University of Illinois campus. Champaign-Urbana, I should specify. And they also have got some prehistoric shells from the Illinois State Archaeological Survey. These were shells that are found in middens or trash heaps of Native Americans, and they're dated to, one batch was dated to 1000 CE, and another batch was dated to around 1200 CE. So these could potentially, and we'll talk about the kind of pros and cons of this, these could potentially tell us what things were like prior to European colonization, kind of give us a background that we can compare our modern and historic archived shells to. All of the analyses that I will be talking about were done with inductively coupled plasma mass spectrometry to look at shell sections. So what I did was I took out sections of the shells and then processed these so that they could be analyzed at the University of Kentucky. And this was to get concentrations across the entire age, the lifespan of the organism. What was it experiencing from when it was just a tiny mussel just starting out to when it was a big old a mussel. And some of these mussels can live a really long time. So typical old age mussels range from 30 to 40 to 50 years old as kind of an upper end or kind of an average old age. Some data I haven't had a chance to process yet. I have done some more temporally intensive sampling in Lake Michigan using zebra mussels. I won't be able to report on that today. I'm still processing that data. And one thing that I'm just going to tell you one thing, part of my approach is there are a lot of elements. You've already seen that I'm going to just kind of focus on 4. I'm trying to figure out how these elements act and see if there are any patterns that I can see and put them in kind of into element families. How do these different elements, do some of them act in a similar way? Do they act in different ways? What could be driving what is going on with these elements? So let's answer some of those questions that I asked earlier on. And the first big question is, can we detect these elements in our shells? And this is percent below detection. So our higher bars mean that there's less detection. You can see that some elements, we were not able to detect them in a regular and consistent manner. Things like aluminum and chromium. Mercury, that's just because we were using ICP-MS for a few samples, we switched that up for cold vapor atomic absorption spectrometry, and that worked a lot better. So some elements, and lead I include in this, I just don't think that shells are always a reliable source for that. I think that they have a lot of issues with detectability. And so my first kind of conclusion that I came to as looking at the literature and doing this is that one family of elements is elements that I'm not sure you're going to have to be pretty careful to draw conclusions from those elements, because I'm just not sure that they are accumulating at high enough concentrations to make them able to be analyzed, although that could again depend on your instrumental method. And you might have also noticed in the previous graph I had some stripedy bars to show you what the meta-analysis said of below detection, and they were almost always lower than what I got. The only difference would be in arsenic and nickel. And I just think that the literature doesn't report below detections as often. I also undertook a really rigorous cleaning process to remove all sediment particles, because trace elements can cling very tightly and at high concentrations to sediment particles. And I'm not interested in the sediment particles, I'm interested in the shells. So I used trace metal grade hydrogen peroxide to clean all the shell segments. And this was not typically done in all of the studies for the meta-analysis. So I think they were analyzing a fair number of sediment particles there in their concentrations. Okay, so that was my first conclusion. As I said, I'm going to kind of try and go through this a little quickly so I don't take up too much time. So I do have the next thing is to connect those environmental concentrations to concentrations in the shell. And again, I do have some data on this. I just haven't processed it yet, and I'm hoping to get that out and published this summer. But my meta-analysis turns out that this has not been done a lot. In my meta-analysis, these are all the elements that where they compared sediment concentrations of the element to shell concentrations. And I only included elements if there were at least 3 studies, those parentheses down at the bottom to show you how many studies actually did this comparison. And then I used a vote counting system. Yes, there is a correlation. No, there is not a correlation. So this has not been done. For the highest number I was able to find was with zinc, and only 10 studies, and this includes all systems, marine, estuarine, and freshwater systems. And for these, the yeses, the number of times that the percentage of studies that did show a clear correlation between sediment concentrations of the element and shell concentrations is still fairly low, even though I mentioned that I think I have a superior sediment removal process. The correlations are still fairly low. And again, if we look at water concentrations, again, it's fairly rare relatively. It's more common to find no correlation between water concentrations and concentrations in the shell based off of the literature. Again, this has not been studied super thoroughly. And so my second conclusion is that I think right now there's weak evidence that environmental concentrations correlate to shells. Well, however, one thing that all of those studies had in common is that they were working with grab samples. They were going out, they were grabbing some water, they were grabbing some superficial sediments, and then they compared that to the shells that they grabbed at the same time. And because the shells represent a long-term exposure to the organism, I think that is probably where that lack of correlation has the potential to come in. The other place where I see a lack of correlation is that I've found no studies that I can recall that looked at suspended particles. Remember, those are the particles that the mussels are filtering out. And so that is probably the main route of exposure for these trace elements. And probably if we could get some robust studies looking at that, and again, over time, I would predict more of a correlation, but you need that. I think you need that time element, and I think you need to see those suspended particles. And I do have some data with the time element, not a super long period of time that I'm using short-lived mussels, but I do have some data with the time element that I'm hoping to look into some more on that. So that's the point where having a project going where you can really look at what's going on in the environment over time and comparing that back to a mussel that's been there that whole time, that's where we could start making these connections. So what did I find? So I found that so far I found that some elements I'm not really going to be able to take a look at. So far, I'm not sure that they can be directly correlated. So let's look at where all of my shells came from. And so I have 2 maps. This study was so large, we split it up into 2 papers, one for the main stem of the Illinois River and one for the upper tributaries of the Illinois River. And you can see on the left, I have a map of the mainstream from the mainstream paper showing the 5 sites along the river that we collected, my colleagues from Illinois Natural History Survey collected mussels in modern times. One of those sites, Havana, also had archived shells that they were able to use from 1897, 1909, 1912, 1966. And that site at Havana is also where the Archaeological Survey had some shell middens. That's actually, Havana, Illinois is actually the site of Dixon Mounds, a famous archaeological site of pre-Columbian America. There's one more site up at the confluence of the Des Plaines and Kankakee River where the Illinois River is first formed. That's where our other archaeological set of shells came from for our upper watershed. So on the right, you can see the map for the upper watershed, and we had 5 sites where we had historic shells. Those were all from the 1950s and 60s, and then 3 sites where we had my colleagues were able to collect modern shells. So this is where our data is coming from. And so what concentrations did we find? So just looking at the means of every collection, a collection being a specific site and a specific time, I start sorting these into groups. And here each open circle is a different collection, and the black bar is the mean for the pre-Columbian background. You can see some elements I can't report a pre-Columbian background because I didn't get enough above detections. Some elements I could report a pre-Columbian background, but I can't report very many modern or historic collections because of below detections. And then you can also see with some of our elements, so for instance, we can start comparing them to this pre-Columbian background. And so for instance, taking my exemplar elements, manganese is typically lower in the modern and historic shells compared to pre-Columbian. Copper is typically higher, and then cadmium tends to be on both sides, both higher and lower than that background. So this is a start. This is where I kind of started, as I tried to think about how these different elements acted. And so our first conclusion, kind of rough conclusion that we can get, is some elements are enriched compared to that background, such as manganese and zinc, or excuse me, such as arsenic, cobalt, copper, and nickel. Some are depleted, such as manganese and zinc. And some are, I say here, similar. I couldn't come up with a better word. They're around the background, or they don't tend to be significantly different than the background. Again, though not in all cases. This data set got really complicated really quick because there's a lot of shells, a lot of locations, a lot of time points, multiple species. So it gets, I'm going to try and keep it as simple as possible. Let's take a look at what we're able to find for some of our elements. Here's manganese. As I mentioned, it is, on average, it tends to be lower than our pre-Columbian background. And at the Havana site, it had a significant decrease with time over the 20th century, the course of the 20th and early 21st century. We think that the high concentrations in the pre-Columbian shells are likely caused by diagenetic enrichment. So just by the fact that they were in the ground, the manganese preferentially either moved into the shells or was not released by the shells over the time that they were in the ground. And this comes from the literature. I'm not an expert on genetic enrichment. That is something that goes a little bit over my head, but looking at the literature, all the archaeological studies that have looked at manganese have found this enrichment factor in both shells and in other, I've seen that in some other things like pottery as well. So this is something that manganese does over time. And so that's likely why it's so much higher than our modern and historic shells. Taking a look at copper, which again was higher than our pre-Columbian background, it also increased in our samples over the course of the 20th century. And the simplest explanation is that, well, we were polluting some more, right? So if you add copper into the system, it's going to show up at higher concentrations in the shells. And there's no evidence for diagenetic changes in copper in shells, although that is, again, something that has not been super thoroughly studied in the literature. And then if we look at our third exemplar, cadmium, no significant difference. At Havana, a weak increase over the 20th century. The p-value is like 0.9 something, and the r-squared is like 0.05 or something in that neighborhood. So a weak increase, possibly, so some evidence of contamination, but not a lot of change. And so there may be limited anthropogenic sources. Cadmium may just be, in certain cases, at least in the case of the Havana site, seems to just kind of be geologic. It's just kind of the same in all of the shells, or relatively comparable in all of the shells. We can also, with this data though, compare what's going on. And so these shells were from 2013. What was going on up and down the Illinois River? So we had 5 sites of the Illinois River. I realized as I was practicing right before this talk, I was like, there's 5 sites, but I only have four points on this graph. So I don't know what happened there. I'll just admit it. I realized I don't know where the other site went. But I do know because I checked this against my papers, we were able to find significant shifts up and down the river for certain elements. So there seems to be higher concentrations at one end of the river than at the other end of the river. Typically, the higher concentrations are at the lower end of the river. So there is some indications that there may be shifts in geology, there may be shifts in human impact as we go up and down the river. Switching gears a little bit and looking at those Illinois tributaries, now this is where it gets complicated. So if we switch to look at those Illinois tributaries, we have 5 different sites, and at only 3 of them were we able to find modern shells. So the Fox River used to have a very large mussel population, and it's more or less depopulated of mussels at this point. And so my colleagues were not able to get modern shells from those 2 sites in the Fox River. So our closed boxes, our closed squares in this graph are the historic shells, all from the 1950s and 1960s collections, and then the open circles are our modern shells. Taking a look at our exemplars, again, there is our background, all right, our pre-Columbian background. For manganese, it is again high, although now we're in the tributaries and the rivers are smaller, the geology is different, the human impact in the 20th century is much higher because this is close to Chicago. And now we're seeing at one site on the Des Plaines River that it's not significantly lower than the background. And I'll just say that we were looking for comparisons. We were predicting that we'd see higher levels of these trace elements in the mid-20th century, prior to the Clean Water Act, prior to decreasing some of these contaminants in the environment. And at Des Plaines, manganese was increased over the historic shells, whereas at DuPage River, it was significantly decreased, and at Kankakee River, there was no significant difference. Taking a look at copper, again, so kind of looking at our exemplars again, once again, all of those sites were above background significantly, but once again, they all behaved differently. The Des Plaines and DuPage Rivers, not significantly different historical versus mid-20th century, but the Kankakee River, copper apparently increased by quite a large margin, significantly in the shells compared to those historic shells. I do think that there is I do have some theories that I talk about in my paper, but this is, the Kankakee River ended up looking very different than the other rivers. If you could say anything about the other 2 rivers, you had to say then the exact opposite for the Kankakee River. We'll see this on the last graph when we take a look at cadmium. Yep, at cadmium, it also increased at the Des Plaines River significantly, whereas at the DuPage River, it really decreased. And at the Des Plaines, there was no difference compared to the mid-20th century. And again, cadmium is around the background, although at some of those mid-20th century shells from the DuPage River and the lower Fox River are significantly higher than our backgrounds. So I do think that that's evidence that cadmium was locally important as a pollutant, but within the whole river system, it's not been much of a pollutant. So how do you take these, and again, these are just 3 of my elements, 3 of the elements that I took a look at. How do we take all of these and get a story out of them? Like a story that makes sense. Like some of them are up compared to history, some of them are down, some of them are up compared to the pre-Columbian background, some of them are lower. What's going on? Oh, I've got one more slide. If we look at those across time, only cadmium showed a significant drop across time. The others did not show a significant change over time. If we throw all of the sites together, which is not really a great approach, but it's what we had. then cadmium did show a significant decrease. So I start trying to figure out, oh, I've got it. Do I have a question? Oh, okay. Thank you. So I'm trying to figure out how to explain what I see. And so I've got some ideas so for instance, some elements I can pretty clearly point to direct human effects, as in human contamination, that can explain some of the results. For cadmium, I think that there's regional pollution up in the upper watershed, but that does not impact the main river. And for copper, it's pretty clear that at every place you go, copper has been enriched and it's changed over the 20th century and it increased. However, I also think there's a chance for indirect effects, and oh, wait a minute, copper's in that category too. So why do I think that there's indirect effects for copper? I've mentioned manganese, probably related to, we thought at first related to dissolved oxygen. So let's take a look at one more piece of data that we used to try and figure this out. For those upper tributary shells only, we looked at stable isotopes. So we looked at stable isotopes, we looked at carbon-13, and we looked at nitrogen-15. And I should point out that this is a bad graph. I was in a hurry to get it together. This is not like manganese causes. I don't want to imply causation here. I know that that's implied, so I'm just showing the relationship. But there is a significant relationship between manganese and delta C13. And this is all of the sites this is all of the sites and times together, but this relationship also holds if you just look at the modern shells and if you just look at the historic shells. So I only considered this significant if I could see it in different groupings across time, rather than just kind of throwing everything together. And so manganese concentrations, at higher concentrations, you see more negative values of delta C13. And as I started digging more in the literature, I found again that this was not widely studied, but there is some evidence. This is from one paper, a very recent paper. There is some evidence that manganese in shells is not as driven by dissolved oxygen as was originally believed. And it may have to do more with what's going on with algae down at the bottom. So primary productivity, which makes it very localized. So that these results are looking at something that is very local and specific to the shells, the mussels where they are in the stream. And up in the upper watershed, as trees get cut down around those streams, it increases benthic primary productivity, and that results in changing the stable isotope signature of carbon in the shells. And that's the best that I can come up with. It turns out that manganese ended up having a much more complex story than I thought. And then the same with copper. Copper looked like a pretty clear-cut case. You know, Europeans showed up, industrialization happened, we started polluting more copper. Copper went up over the course of the 20th century. Copper is higher than our pre-Columbian background. And yet, when we look at those upper tributaries, we see a link with nitrogen, N15. And the work of my colleagues at Illinois Natural History Survey also found that there was a link between N15 and the growth rate of our shells throughout the system. They were doing kind of a parallel study on those growth rates. And what we think, this is a little bit stronger, what we think is going on here is that as nutrient enrichment has happened in these streams, the growth rate has increased. And as the growth rate has increased, I think that there's more organic matter being incorporated into the shell. This is something that I would have to really test out. This is my theory right now. And as that matter is being incorporated in the shell, we know from past studies that copper tends to bind really closely to that organic matter. And if more organic matter is incorporated into the shell, then we're going to see a higher copper concentration that's going to match that enrichment of nitrogen 15 from a nutrient enrichment from sewage and other human sources. So that is, again, a fairly, what looked like maybe a fairly simple story, getting massively more complicated and starting to get outside the scope of the ability of this study to look at it. So my main conclusion is that trace elements in shells likely reflect complex impacts of human managed environments. I'm casting a really wide net here for this conclusion. There's a lot going on potentially, and there's a lot of room to ask new questions and perform new studies to try and answer and narrow down what is going on. I've got lots more slides, but I'm gonna, like I said, I was gonna try and go through kind of a bare bones, if you could call it that, of the study, and then just go ahead and take questions that anybody has. I don't know if you all prefer if I stop sharing my screen and I could start sharing it again if I need to go a slide, or if I should just leave slides up. What do you all prefer?
Thank you, Dr. Wilson, for the wonderful talk. Yeah, so please feel free to ask questions. Use your mic to ask questions, or you can write down at the chat window. I can like read it out.
Hi, Aaron. I have a question. I have a couple questions actually.
Yeah.
I'm at the Geological Survey. Yeah. So yeah, I have a few different thoughts going through my mind, but the first thing is, did you find much differences with species, between species differences that are significant, or were they all kind of plotting overlapping?
That's a great question. That's my very next slide. It's just one that I left out. So when we took a look at, so along the Illinois River, we had, for that first part of the study, we only looked at 2 species. That's the three ridge and the maple leaf. And we found no elemental differences, no significant differences between those 2 species. There is a caveat on that upper watershed data, in that we had to make use of, I think, 3 different species, just because of trying to get the archival and the modern species to match up. And we did find at one site from the Kankakee River, we collected both muckets, Actinonius ligamentina, and wardybacks, Quadrula pustulosa, and we found 3 elements, copper, zinc, and cadmium, were significantly different. Based on this, I've mostly kind of put all the data together species-wise, but I do think that there is that one result does kind of open the door a little bit, that there may be room for more research into the different co-occurring species and whether they deal differently. Now, the big thing here is that all of these species are unionids. They're all long-lived, they're all thick-shelled, they're all very close together. And if you started comparing to other bivalves like zebra mussels or invasive corbicula clams, I would definitely not lump those in and say that, and just kind of make have more of an assumption that those are similar, because their shells are gonna grow differently, they're gonna grow more quickly, they're gonna have different shell structures.
Yeah, I have one other question.
Yeah.
When you're looking at the background, was the background the 1900 time, or was it the earlier time? And if it was the 1900 time, wasn't that also a time of pollution? I guess I'm trying to picture what the pollution source was, and over time, like cadmium, I have no idea where cadmium came from. What kind of pollution source did cadmium come from, and what was it like back in 1900 versus today, and over time, that sort of thing.
So the background, what I said is the background, were the pre-Columbian shells from Native American middens. So all of those shells would have been, the organisms would have been alive around 1000 to 1000 CE to 1200 CE. So those are much older organisms, and they stopped collecting trace elements, except diagenetically, a long time ago. So that's what I said is the background. Again,
Okay, yeah.
not infallible, because as I found in the literature, certain elements will change over time. It's just kind of a known thing. Yeah.
Yeah, it'd be interesting to kind of try, I don't know how, if you have anywhere we can get really detailed, like every decade a shell collected.
Yeah, I
That'd
know. That would be really interesting to expand the time period. I think that they
Yeah.
did the best that they could, and they were it's kind of you're kind of constrained by what people did in the past. So
Yeah.
so it's funny when if you look at the dates that we have for these the shells from Havana. So we have some from the 1800s. Oh, let's go back to the map. It's a little clearer. So we have those like clustered together from like 1897 to 1912. Well, if you look at the just information on freshwater mussels in general, that was a time of a lot of activity. I think it was like one guy who was really into mussels and did a lot of work. And then there's a period of where we don't have a lot of data on mussels. And then in the 60s, there's more interest. And then you see we have a lot of shells from the 50s and 60s. And it's also regional. So some researchers or shell collectors would be active in one part of the state at a certain particular point of time and then not in another. But it would be really cool if we could find more, kind of build that out. Absolutely.
Okay, thank you.
I'm going to go ahead and jump in because,
Yeah.
so my name is Isabel Lelowitz. I'm with the Illinois State Archaeological Survey.
Oh, awesome.
I'm a zooarchaeologist, actually a fellow UGA PhD grad too. So go
cool.
dogs.
Yeah.
And I actually specialize in invertebrate analysis, stable isotope analysis. Most of my work has been done on coasts, down off the Georgia coast and Gulf Coast of Florida. But I'm here in Illinois now. Right. And looking to really branch out and try to get into these terrestrial bivalves. Not terrestrial bivalves, but terrestrial gastropods, and then riverine bivalves, and looking at how people, I'm paleo-environmental reconstruction has been a lot of my work. So here I am. Right. I think that really expanding some of those pre-colonization data sets would be really cool. Yeah. So my question for you is, do you think there are trace elements that can be instrumentive in looking at human landscape management practices that, are there going to be signals that we can see in these pre-colonization shells?
Oh my gosh, I think so. I absolutely think so. Now, if I had to pick an element, would be hard to say. So the real, as I mentioned, so first of all, this, to the best of my knowledge, when we published this study, it was the first time that stable isotopes of carbon and nitrogen had been matched up with trace elements in freshwater shells. Yeah. There's been a lot of more work with oxygen. Yes. So to match that up within the shell to say, okay, well, this represents temperature. So it's telling us that then this trace element happened in July-ish of this year, right? And so that's been a lot more common. I do think, and I'm just going to kind of base this off of, there's been some really great work in marine systems, and they have found been able to do some really cool reconstructions. Actually, I think I have a graph from one of those papers.
Yeah, so most of my work, I've actually done a lot of geochemistry for past sea surface temperature and salinity changes, and then seasonality studies as well. But it's a more complicated, obviously, in riverine systems. Just sort of getting a handle on where, what those oxygen isotopes are actually telling us. Carbon is a lot more complicated. The few studies that have been done in terms of isotope studies for riverine, bivalves, carbon seems to be a lot more complicated. But again, those studies are very minimal. So
Right.
that's one thing that I want to, also try to work out as well, is if we can use some of these bivalves for a little bit better paleo-environmental data.
Yeah. And so what I would say is, I think, so what's typically done is lead is typically seen as a really good indicator of human activity because it's global and it represents it also represents industrialization at all stages, even the early like Iron Age industrialization, and it also becomes global. So as you probably know, cores of sediments will represent peaks of lead due to the Roman Empire, and they've taken these cores from like the Arctic, right? The thing about the freshwater systems is I'm not sure that lead is available enough. I think in a freshwater, this has worked really well in marine systems. This is an example you're probably familiar with from Arctica Icelandica shells out in the North Sea. I'm sure you know the work of Bernd Schoen. Yeah, and so this is one of his papers. That's been really successful in marine systems, but I'm not sure the chemistry is right in freshwater systems. I just don't think it's bioavailable enough, and I don't think it enters in this shell as enough. I do think, however, because I have published a number of papers on this, and I've seen a lot more evidence for it in the literature, I do think that manganese it's really easy to see in shells, and there's a lot of interesting links, again, with dissolved oxygen and primary productivity, two things that are going to be related to land management techniques at all stages of human history. And manganese is, if I was to pick like one that I think would really show a signal, and especially if you could line it up with, because in the past there's been some questions about, well, is it showing temperature? So if you also had stable isotopes of oxygen to help you out with that question, then you could see what else manganese was telling you. Other elements that I'd like to see more on is copper and zinc. Zinc is, I don't know what zinc would show, but zinc is really easy to analyze in shells, and it's showing something. I don't know what, but it's showing something. And then I do think that copper is showing land management. I think it's showing enrichment. I think it's showing growth rates, but I don't have a lot of strong evidence for that. I have some weak evidence right now. So that's what
the link that was copper and the link between copper and nitrogen, correct?
Mm-hmm. Yes.
Yeah. So I think that is going to be would be really cool for the introduction of wide scale agricultural practices,
yes,
like the introduction of maize and
yes,
so I think that would be really awesome. So my field station is in Collinsville, so we're in the American bottom.
Oh wow, yeah.
So we have a lot of that data and that potential data set. So I'm glad I popped on today because this is
awesome.
I've got a lot of ideas, so I may email you later.
Thank you. I'm so cool. Well, I'm glad I'm talking to you. I hope I wasn't. I hope I wasn't like, I was probably just telling you stuff you already knew.
No, because I haven't done anything with trace metals.
Gotcha. Okay.
And I'm very new to these riverine systems and freshwater mollusks. So it's really, this has been great. So thanks.
Awesome. And thank you, because I'm the opposite. I haven't done a lot with, this was my first stable isotope paper with any stable isotopes in it. So yeah, so and thank you for giving me some of that background on that.
Aaron, I just want to add one other thing. If you were ever interested in looking at really old stuff, I don't know if it'd be of importance or not, but we do have mollusk shells of some of those species back into the Pleistocene.
Right. I've read a few papers that have looked at Pleistocene shells, and that's interesting. The concern that I've seen a lot of those shells is that diagenetic change that happens during fossilization, and how to kind of account for that. And
sure.
Yeah, I don't know.
I just thought I'd let you know in case it was ever interesting to kind of see if it's changed through time.
Well, yeah, that's like a deep time background, right? Like that's even even further back. What are we different levels of background?
And whether the background is a constant
Yes, absolutely.
or fluctuating, yeah. But
I think we have
yeah,
a
anyway,
really
also
huge
take
paper
ourselves to
here.
Kevin's yeah, right. Long.
Yeah.
Yeah.
Well,
Aaron,
good to know
what,
that you're there, Isabel. Yeah, we might stay in touch.
Yeah. Sounds great.
Aaron, what parameters or indicators do you have for diagenesis? Do you have a technique to look at things and say this has been altered or not?
I that again, I think I try, I do not know anything about diagenesis. That is, and actually as I was prepping this, I was like, this is a good chance to put this in front of people who really know this concept. As I was going through the literature, I looked for papers that had looked at trace elements from archaeological shells. I found some really interesting ones, like Isabel, there's some really cool ones where they were, because shells were used to temper pottery, they were making their own shell tempered pottery and comparing trace element concentrations before and after firing to see if that caused changes. But all I know is what the literature tells me about that, Neil. And so any insights that you all have about that would actually be really helpful, because that's just that's outside of my my area. I was just kind of relying on what other people had said, and they said manganese definitely changes over time. Other ones not so clear.
Okay. I know that in the marine foram literature, where they like to do the Delo 18 on the forams and sediments, they worry about diagenesis a lot because that can reset the isotopic clocks. And it's been years ago when I saw this, but someone actually was using FTIR, which I'm sure you've got instruments in your chemistry department, where you could look at the mineralogy of the carbonate.
Oh, cool.
And so you can tell the difference between aragonite and calcite. And my guess is the peak width probably changes with crystallinity.
Yeah.
And so there's probably some little metrics that one could develop by looking at a time series that might give you an indicator of diagenesis.
I know, Neil, like for the carbonite, the radiocarbon, I know they often look at the SCM images for the gastropods to see if they're recrystallized. I don't know about the manganese at all, but yeah.
Right.
Yeah, that's what I was just going to say, is that one of the first steps that we will take is doing SEM images to look for dissolution. Dissolution is really easily characterized in fresh breaks. And so that's one thing. That's definitely somewhere we always start when sampling. And if someone does decide, I have a whole soapbox on doing radiocarbon dates on shell, but I'll do that here. But it is one, definitely one step if you are gonna do radiocarbon dates on shell, is to look, do SEM. Yeah.
Yeah, I think I can't remember how those shells were dated. I just took the dates that were given to us by the archaeological survey, and I was like, all right, this is our basis, because that's I just kind of relied on them to do best techniques.
Yeah.
Any other questions? Thank you all for joining the webinar. Thank you, Dr. Aaron, for joining us today.
Thank you. I want to thank you. I want to thank Tarun for organizing this. He made sure that I got all my materials in on time. He did a great job.
Thank you. Lala, do we have any other announcement?
No, I think we're good. Thank you so much, Dr. Wilson, for joining us. And
Thank
I
you.
think this was a great discussion too. We were happy many people who were interested in this topic was able to join. So thank you so much, everyone. And the recording will be up in our webpage in YouTube. So we are gonna share the link later today. Thank you so much, everyone, for joining.
Thank
Great,
you.
thank you.
Thank you very much.
Okay.
I can't really write